So today's webinar is entitled A Breakthrough in FFPE Analysis and Next Generation DNA RNA Extraction for NGS applications. And I'm happy to welcome the CISO and also co-founder of Purigen Biosystems, Clint Rose, who will be the presenter of today. Clint, we appreciate your presence this early in the morning, your local time in the US. And just a little housekeeping before we get started. You are all muted by default. So if you have any questions, please type them in the chat box below, and we will gather them. And after the presentation, we will have a Q&A session, and we will address as many questions as possible. We plan to have a Q&A session of about 10 minutes. If not all questions can be answered, we will contact you later on. The presentation will probably take about 20 to 25 minutes. And now I would like to give the floor to Clint. The floor is yours.
Great, thank you. Let me just get the slides up. Go. Okay. Thank you everyone for joining. Excited to talk with you today about the system and the kits that we've developed for helping you extract DNA, RNA, microRNA from your FFPE samples in preparation for going into NGS analysis. I think anytime we talk about FFPE, it is really helpful to talk about the sample itself. And remind ourselves that it was never intended for this type of analysis. It was actually intended for microscopy, the ability to harden that tissue, preserve that tissue, actually preserve that tissue for decades, maybe even centuries. But what it's provided for us is the ability to have this time machine. We have captured that DNA and RNA in these samples, and we can go back and utilize those samples to dig deep, really start to understand disease states over a long period of time. What that means as we start thinking about the genomic applications, though, is that there's a ton of archives, there's opportunity to really go back in time, but that is at the expense of nucleic acid that's generally pretty heavily degraded. So we just have to think carefully about how we're going to extract and how we're going to use that nucleic acid. And of course, there's cross-linking. And so anytime we're thinking about how do we get access to that nucleic acid, we have to think about those cross-links. How do we remove those cross-links? And we've found lots of good references that I think really help focus in on what the real problem is when you're dealing with FFPE samples. Across all three of these, what we're seeing is that about a third of FFPE samples actually fail QC for downstream sequencing. And if you dig a little bit deeper, it really just is as simple as either you can't get enough yield, and generally as we think about sequencing assays, there is some minimum amount, say 100 nanograms, 200 nanograms, of DNA or RNA that need to go into that analysis. And so you're either failing on that yield or you're failing on quality because the DNA or RNA has been either heavily degraded as it was stored in the FFPE state, or because the extraction process is not taking good enough care of that nucleic acid during the extraction. And so with that in mind, both with the idea that these are not normal samples, and the fact that we need to get as much as we possibly can and preserve whatever integrity of the nucleic acid is in that block. That really guided us in terms of how we have developed the system and the kits for doing FFPE extraction. So what does that mean for you as a user, potential customer? What that means is we want our process to be as simple and robust and automated as possible. FFPE is generally really challenging to work with. And so we have worked hard to make sure that we minimize potential for error, that we make it as hands-free as possible. We want to minimize any complicated steps as much as we possibly can. The second thing is really optimizing yields. As you saw on the previous slide, getting over that minimum amount is really, really important so you can do your downstream analysis. And the kits that I'm going to talk to you about today, between our DNA kit, we get about 3.5 times more DNA out of FFPE samples on average, about twice as much mRNA. And if you're interested in microRNA, the kit that we use for RNA will also co-extract the microRNA and get about 200 times more than even the kits that are specifically designed for microRNA. And then the last piece, it's really about improving the quality of the extracted nucleic acids. I will point out, we're not trying to repair the nucleic acid. We're not doing anything to chemically change the nucleic acid. What we want is we want to maintain the quality of the nucleic acid as it exists in that FFPE block. And as I'll show you in the slides coming up, the main thing for us is we do not want to change the information that exists in that block for you. We don't want to create any bias. We don't want to lose material for you so that you can get the best quality data on the back end. So this is the system that we've developed. And as I said, everything is designed to be as simple as possible. The system itself is goes on a benchtop. It's about the width of a standard laptop. So intentionally trying to take up as little space as possible, especially in the busy labs. Really, really easy to use. All of the protocols are already on the instruments or can be updated to be put on the instrument. It's fully automated. It will guide you through each of the workflows. And each of the kits that we have come with an application-specific microfluidic chip. That's what you see in the upper left there, as well as a set of chemistries that enable you to perform the Isotachophoresis unique separation that I'm going to talk about. So let's dive into how our technology is different. So when you think about traditional sample prep, whether you're thinking about beads, whether you're thinking about columns, that's basically the same approach. Those are just utilizing solubility and therefore chemistry to bind your nucleic acid onto a surface, whether it's the surface of the bead or the surface of the column, washing across that surface, and then changing the chemistry again so that that nucleic acid will elute back off that surface. So I'm sure everyone has used either beads or column. You know that generally they work. You can get nucleic acid, but because it's surface-based, what happens is you can get loss. You can have material that never binds, never releases back off of that surface. You can get contamination in terms of whenever you're doing that wash step. You also are risking bias, and I'll talk more about that in the slides coming up. So ITP is completely different in that we're taking advantage of the inherent property of nucleic acid, which is that it is incredibly highly charged. Also, not only is it highly charged, but how fast it will move in a field, which you'll see is incredibly important, is not dependent on its length. And so we will take advantage of those two things. What this enables us to do is to do a solution-based extraction. Because we never bind to a surface and we never have to worry about getting the material back off of the surface. So we're going to get you high yields, high integrity. We're not damaging the material as it goes through. Isotachophoresis is also a separation technique, so it is inherently going to give us high purity. We can remove the nucleic acid away from any inhibitors within the system. And in the end, and what's most important as a user, is we want to give you the best representation of your sample. As I said, we do not want to create bias. We do not want to lose material, change the material in any way so that you get the best quality data. So let me dive a little bit deeper over the next couple slides to just explain ITP to you. The way that ITP works is it is primarily based on two buffer systems and really just two ions. The first is what I show you in the blue. These ions are what we call leading ions. These are just fast ions that we've selected, and we want them to be faster than nucleic acid. And I can tell you, nature sort of selects that for us because chloride is our primary fast ion, and it comes with all the biology. So it's a very nice, easy choice for us. The second ion that's really important is what we call our trailing ions. And those are ions that we select that to be just slower than the nucleic acid, but we want them to be faster than all of the impurities in the system. Now, the other important thing that we've done is we've developed all of our own lysis chemistries, and we've developed those lysis chemistries to enable us to lyse, to gain access to the nucleic acid, to remove crosslinks in the case of FFPE, so that we now have free nucleic acid within a buffer that is immediately compatible with the ITP process itself. We then use our microfluidic chips, as I'll show you on the next couple slides, to then line up all the buffers in series. We want these slow ions in red in our cathodic buffer sitting in the back. Then we're going to take our lysate that has our nucleic acid, put that next. Then we're going to have our separation buffer that has these fast leading ions in it. And then last, really important, we have our extraction buffer. And our extraction buffer also has these fast leading ions in it, just at a very low ionic strength, so that that buffer is immediately compatible with all of your downstream assays. Once we blind everything up, we turn on an electric field, and at that point, it just becomes a race. All of the nucleic acid will move very fast in the electric field, but it can never speed ahead of these blue ions because they are faster. It is, however, faster than all the red ions, and so what you get is this stacking effect where all the nucleic acid will end up in a very tight band right in between the leading and trailing ions. Now, as I mentioned, nucleic acid, very highly charged. And what that means is that it is faster than the impurities within the system. And so those impurities are so slow that they're actually dropping back behind the red ions. So you're getting separation and purification at the same time. Now, this process continues until the nucleic acid arrives in the extraction well. At that point, the system actually detects its arrival. It will shut off the field for that channel, and then you can come in and pipette back out that purified nucleic acid. So that is the cartoon version. Let me show you what that actually looks like. This is a view of one of our standard chips where we are seeing 8 lanes running in parallel. In this case, I'm showing you dyed genomic DNA, generally no dye in the system. This is just a nice visualization. And you can see that ITP effect where all that nucleic acid gets compacted onto that tight band. As it's moving to the left right now, this is where the purification is happening. And if we now overlay back in the cartoon I showed you on the previous, you can see that process in action. All that nucleic acid going onto that interface between the blue ions and the red ions. And because it is so fast and because the impurities are slower than the red ions, they are dropping back behind. That's where you're getting your purification. The process continues, as I mentioned, and will go on until the nucleic acid arrives in the extraction well. And at that point, system detects its arrival. This is the point where you can come in and pipette out. So as I mentioned, one thing that's really important for us is to make this as simple as possible for the users. And so this gives you a sense of once you have a lysate and you're at the instrument, it's as simple as loading the running buffers. You can see multi-channel pipette to enable this. So it's 5 pipetting steps in order to put the buffers into the chip. The instrument will then prime the chip for you. So it's putting all the buffers into the correct location, opens back up one time. That enables you to put your lysates in. At that point, you simply tell it go. And about 50 to 60 minutes later, you'll have your purified nucleic acid. At that point, you can come in, pipette out that nucleic acid. So now I'm going to jump into each of the individual kits, talk to you about some of their data, but I think it's helpful. Let's start with the FFP to Pure DNA kit and just take a look at the workflow. So up top is a typical column-based workflow, and you can see the difference in terms of how we do it on the bottom. You simply start with your scroll or start with a slide and scrape that into a tube. We then add our lysis buffer. Incubate that. We recommend incubating on a thermomixer. That enables you to control time, temperature, and actually some RPMs so that you can shake just a little bit. That process then becomes hands-off. So you're lysing, you're removing your crosslinks, you're getting that DNA ready to go into the purification step. Once that's completed, then it goes on to the instrument, just like I showed you on the previous slide. And then once the purification is complete, you have ready to use DNA. Right, so I'm going to talk to you a little bit about data, some of the data that we've generated using the FFP to Pure DNA kit. Here I'm going to show you 32 unique blocks. For those of you that have worked with FFPE, you know every block is unique. And so it is really important to do head-to-head comparisons on a block-by-block basis, and I'll show you some data to go along with that. So here is an example of the amplifiable yield. So actually using a qPCR target and looking at the amplifiable yields across 3 different tissue types. And as I said, 32 different blocks that we're comparing. You can see the Ionic System here in the blue compared to the column-based kit in the red. And across all of these blocks and across all the different tissue types, you can see it's about a 3.5 times improvement in terms of the amount of DNA that we're able to get. I will point out not just DNA that's coming out, but usable amplifiable DNA that's coming out. Second really important thing is to ensure that we're getting high quality. This is an assay that we like to use called the QCplex assay. And this assay is basically telling you, is my material amplifiable? There are several different lengths of target that are in this assay. So it's also giving you a sense of how high of the quality of that DNA that you're getting out. And you can see for the exact same 32 blocks I showed you with yield on the previous, we're also getting much better quality out of those blocks as well using the Purigen system. Now, the next important thing is what happens when we actually get to the sequencing. What we've found is in many cases with the column kit, you can actually get some amount of bias. We're looking at a coverage ratio here, where we're actually looking at how much coverage we're getting on each of the targets versus the expectation being a very high quality DNA. What you can see is there is no bias. We're basically getting all the target hits that we expect with the Ionic System versus length of the particular target, whereas with the column kit, you can see there's some bias out here as you get to longer length targets. And the same thing actually happens with percent GC. So with the Ionic System, there is no GC bias, whereas with the column kit, as you get to the high GC content, you can actually start to see some bias there. Now, this starts to really matter whenever we start thinking about things like looking for variance within a sample. This is a simple study that we did with the exact same block going into the Ionic System versus a column-based kit. And here, what we wanted to see is if we put in 1, 2, or 4 5-micron sections into the extraction, what does the sequencing data look like? And what we found is that for the Ionic System, very consistent in terms of our coverage, we were able to detect 25 out of 26 with a single 5-micron section, and all of the variance within this sample whenever we had 2 or 4 of these 5-micron sections. Unfortunately, with a column-based kit, this is just not the case. What we see here bias, we're actually losing information on particular targets, and that translates into an inability to actually call all of the variants. You can see even out here with this particular sample, where there were 4 or 5-micron sections going in, only 23 of the 26 variants were found from that particular sample. Right, now I'm going to transition over to the FFPE to Pure RNA kit. As I said, this will enable you to get both mRNA and microRNA from a single workflow. Very simple and very similar to what I just showed you for the FFPE DNA. You're simply dropping it into the tube, incubating, and then you're ready to purify and collect. So for the mRNA case, this is a comparison looking at several different blocks, several different tissues as well. 10-micron sections into each of these extractions. And what you can see is on average, we have about twice as much yield as you can get from the market leading kit, whether it's a small, fine needle aspirate tissue or a much larger resection. Also really important is the quality. And for quality with mRNA, we like to use the DV200 metric. I will point out DV200, as you probably know, is measuring how much material you have that is greater than 200 nucleotides versus the total amount of RNA that you have. And so because we're carrying forward from some microRNA, that will tend to bias our numbers down just a little bit. Despite that, what we find is we're generally on par with the DV200 scores that you were able to get from a standard column kit. And you can see that across all the samples that I just showed you. Now, I also mentioned you can get microRNA. This is an example where we're looking at 2 specific microRNA targets across these different samples. And what you can see is the difference can be quite dramatic. In this case, several of these actually have no microRNA detectable coming out of the microRNA specific column kit. That's what you see down here in the dropouts. For those that did have some measurable amount of microRNA, what we find is that our kit was able to get about 200 times more microRNA across these different targets. So next, I want to talk briefly about our FFP Complete Kit. What this kit enables you to do is actually get DNA, RNA, and microRNA from the exact same starting scroll. Now, what we do for this particular kit is we actually, because we have such a yield advantage on both the DNA and the RNA, we actually drop the scroll or the slide in, we lyse, and then we actually split that lysate, and we're going to do two separate extractions. For one extraction, we're going to add DNase so that we can collect RNA. The other, we'll do RNase so we can collect the DNA. Now, the advantage of this is one of the significant advantages is in the automated workflow. You can see a comparison here against two of the standard kits and the amount of hands-on time. So we've taken a hands-on time that can basically take an entire workday, 6 or 7 hours, and simplified that down to just an hour and a half. Now, again, we are splitting our sample in half. So the expectation is that we're giving up half of the DNA or half of the RNA. And yet, despite that, from an mRNA perspective, we're able to get about 20% more RNA than you can from your standard kit, from your standard column kit. Left-hand side, I'm showing you the qubit, so the total RNA amount. Right-hand side, I'm showing you a specific target, so that's your amplifiable RNA. And again, 1.2x improvement in both cases. For the DNA case, what we find is we actually have two versions of this protocol. We have what we call our quick DNA protocol. That is ready at the same time that the RNA is, so just about an hour. In that hour, if you pull that off, go ahead and run your DNA. What you find is that the DNA yield, total yield from qubit, or the amplifiable yield is comparable to what you can get from the column-based kit. We can further improve that if you want to take it and take that DNA back, incubate overnight. By incubating overnight, what we're enabling is we're removing more of those crosslinks. We're getting access, even more access to more of that DNA. And once we go overnight, what we found is that we start to get an average yield improvement via qubit of about 1.2x. And the real dramatic improvement is on the amplifiable, where we found that we got about 2.5 times more amplifiable yield once we take that DNA back through. And then to finish up, you've seen the excellent results that we get from fixed tissue from the FFP samples. We also have relatively new products that enables us to go from fresh tissue to pure DNA. These are fresh frozen and very similar story. So in this case, we looked at a broad range of tissue types going in. In each case, we're putting in a fixed amount of tissue into these samples. For those of you that work with fresh tissue, you know that the metric is a little bit different in that you want to represent your yield as the amount from a mass perspective of DNA that you get out versus the mass of tissue that you put in. So micrograms over milligrams. And what you can see is that even when we compare against these two kits, there's kit A here and kit B. For kit B, we can either elute into 50 microliters or 400 microliters. We wanted to show that doesn't matter which of the kits you're using. It doesn't matter whether you're eluting into a small volume to preserve concentration or a large volume to try to get as much yield as possible. In all cases, the Purigen kit is able to get you much, much more DNA yield. We have high yield tissues over here. We're able to get up to 2 micrograms and sometimes even more per milligram of tissue going in. And these low yield tissues over here, ones that tend to be a little fattier, where we're able to get consistently higher amounts of DNA back out for the amount of tissue that's going in. And on average, it's about a 3.5-fold increase in the particular yield. Great. So with that, I'd like to conclude. Happy to answer any questions you may have about the Purigen system or any of the kits we have available.
Thank you, Clint, for this this nice presentation, which in my opinion clarifies the potential benefits of this Purigen platform. Yeah, we have some time for questions, so please feel free to type your questions in the chat box, and we will try to answer them. Okay, I see already a question from Gerard. Question is barcoding tracking of the sample possible? Could you comment on this, Clint?
Can we get a little bit more explanation from the question? Is it barcode tracking, meaning inputting information about the sample into the system?
I guess it's a kind of sample track and trace related question.
Okay, so what we can do, which is very helpful, is we don't put a ton of information about the samples into the system. However, as you're entering and you're working through each of your workflows, what you can do is you can go in and you can label each sample, so you know A through H what you're putting into each of those samples, and that will get logged. So you can give a generic name, you know, sample with a date and lane A, or you can call it out very specifically. It can be FFPE block C, sample 1, things like that. So you have the ability to actually put more information in about each of your samples. I will also point out one of the benefits, we don't talk about it much here, is that there's a ton of information because of the automation about each of the samples themselves and how they're running and how they're performing. And we're able to see all of that. And so we can utilize that to help troubleshoot. If you're seeing something that looks strange, you get a lower yield than you expect for particular samples, we're actually quite good at being able to identify what the issues may have been. The other benefit we get is if a customer makes a mistake for some reason, puts the wrong buffer in the wrong place, we can actually also identify that from the logs that we're able to get.
Great. So that means there's kind of sample track and trace in regarding a log file as being created every run.
That's right.
All details can be pulled out of the system.
Yep.
Okay, thank you. So there's another question from Jonathan. He's asking, what's the maximum size of DNA and RNA of the fragments you can get out of the system?
It's an interesting question. I'm going to assume it's a Oxford Nano based question, because that's usually where people care about how long the material is. I will answer the question in a slightly different way, only because we're primarily talking about FFPE in this instance. From an FFPE perspective, length is almost exclusively controlled by how the material was fixed. And generally what we find is a lot of material is going to be relatively short, just inherently from the FFPE fixation process. In some cases, we've worked with groups where they are able to get relatively long material out, you know, tens of kbs out from FFPE, but that is definitely more challenging, and more challenging from the sample type, not because of a limitation of the instrument itself. I will say, generally, we are preserving, especially from the FFPE case, the inherent length profile of what's coming out of the block itself.
No, thank you. Another question, is the procedure applicable for other type of samples, for example, blood, etc.?
So that is a type of sample that is in the development pipeline. So we don't have a kit right now for working with blood samples, but it is a type of sample that we're very interested in. So expect to have something like that in the future, but not for the kits that we're selling right now.
Well, another question from Leon Lebrun about pricing. Leon, I will comment on this. I will be happy to contact you about this and share the pricing details of both the instrument and the sample, the kit. and reagents. So I will get in touch with you. Another question about the price. So everyone who's interested in price, please contact us, and we will get back to you. Another question from Ronald van Eyck: are you planning to develop a TNA kit? So DNA RNA combined.
Yeah, so we don't have an official TNA kit. We have been working with some customers to test out, basically just not including the DNase or RNase as we do the FFP Complete extraction. And so if you're interested in that, that's an opportunity where we can potentially do some testing with you to see if it works well for your particular application.
Okay, I think for now that were the questions. So if there are no further questions, maybe Clint, you want to address something, and otherwise we can wrap up.
Yeah, I just to expand on what my previous comments. For those kind of unique samples, if you're not quite sure in terms of your sample, please reach out to GC Bio. You can also reach out to Purigen. We do have an internal lab that works on some of these unique sample types that will help us enable you in terms of testing out to make sure that it fits your application.
Yeah, I just want to add that we have a system installed at our laboratory in Waddinxveen, and if you're really interested to test your sample on this platform, we will be happy to offer you a kind of proof of principle test. So we can arrange, you can send some samples to us. We will do a run and send back the purified material to you so you can analyze. So feel free to contact us about this if you're interested. Okay. Well, Clint, thank you for your time, and thank you for my colleagues who arranged this, from the marketing team who arranged this webinar, and everybody. Thank you for joining, and have a nice day.